Yes, definitely maximize time for this tutorial. But before I get to that, I did want to thank everybody for attending this symposium. I hope it was interesting and informative, that maybe you got to meet with some new people, learn about new research, and that's a big part of the goal, the community-oriented goal. I will be doing a tutorial for those of you who want to learn more details on how to do some of the stuff. It'll be about an hour, and it focuses on more of the higher end informatics and analysis tools. Thank you also to the advisory committee for the CyanoCyc that Peter pointed out, that we've been working with over the last 8 months. It's been a really wonderful collaboration, and have enjoyed that tremendously. And also to Danny Ducat and Himadri Pakrasi. I don't know if he's still on, but both of them were obviously instrumental in getting this CyanoCyc idea going, and I appreciate all the support that you guys have provided to having this move forwards. And with that, I think I will switch over to the tutorial, and so stay on, and I'll be in touch with everyone afterwards with a survey link. So thank you very much. And let me get my screen ready for the tutorial. Okay. It does not look like it's working. Do you see that? No. Let me stop again and try that again. I don't know why my screen has trouble with this.
I did see your screen, Lisa.
You did? Okay.
Yeah.
Usually I see a green outline, but okay, let me try that. Do you see the PowerPoint?
Yeah.
Great. Okay. Excellent. Let me, okay. Oh, that's, don't need that. Okay, so what I'd like to go through today is very briefly some navigation basics for people who've never used BioCyc, just to show you how to move around and the structure of the different information pages. But then I want to focus on 3 categories of tools. So the SmartTables, which we've mentioned a couple times, what those are and how to use those, and some of the things that you could do with them, because they're very smart and very useful. Also comparative analyses, which I find incredibly useful to look at things across different organisms, across the organisms within the BioCyc collection. And then omics analysis, so that you can work with your own data in combination with the database, the organismal databases. So I'm going to use the, I'm not really going to be going through my PowerPoint that I have here. Rather, I'm going to go through it on the internet, on the website. I'll be making my PowerPoint, and we'll be providing the video of this symposium available online or afterwards. But want to point out that you could either start in cyanocyc.org, which is the entry point to CyanoCyc, or you could just start in biocyc.org. Either one provides access to all the databases. And also point out that best to log in so that you have, and logging in is for with your account. I already am a subscriber through SRI, so the account is different. And I'm going to now hop out of my show and go to CyanoCyc here and make sure, let me know if you can't see that. But so this is our CyanoCyc portal, and just to scroll down, we do have an information page that we just put on that gives a lot more information, links to the lists of cyanobacteria based on phenotypes, how to learn more, how to engage with us, etc. So if we go back to CyanoCyc and note that when you click on an organism name, it actually takes you to a summary page. So if we go to this tool, the blue bar up at the top and the toolbar, Peter showed you some of a list of the tools, and they're all basically here. Summary statistics page is the home page for any organism, and on the page it indicates where the genome information came from, and usually if it's curated, you have a little summary about the organism as well. Okay, so navigating around, you can change your current database, clicking on this, and it comes up with a sub-window that allows you to either just start typing in the name of an organism you're interested in. You can search based on taxonomy, and that is the NCBI taxonomy, as we've mentioned, and cyanobacteria fall under Terrabacteria, which then is where the Cyanobacteria melanobacteria group lies. So you can look that way. If you have, when you go in with your account, a preferred database can be chosen, and any recently used one can show up as well. So that makes it easy. Also, with respect to the organism that you're looking at, we talked about synonym changes of taxonomic names. We've tried to provide synonyms for as many of the cyanobacteria as we know of. And you can, so hopefully that'll help with finding things. All right, so we'll just go back to Synechococcus elongatus. And the other thing is quick searches. So for navigating, you want to go to some information page. And so you can navigate here by using a quick search, and you can enter gene names, protein names, pathway or reactions, EC numbers for the reactions, etc. And I'm going to put in, I'm trying to see what I was working. Oh, yes. The gene that I'll focus on is a subunit of Rubisco, and CBBL, and this gives you an idea of what a gene protein page looks like. The structure of the pages has basic information at the top with synonyms, any accession numbers. And in this case, this gene is an enzyme, so it carries out some reactions, and those reactions fall within some pathways. To the right of most information pages are operations table, which provides many other tools that can be used to analyze or browse aspects of the information page. These operation panels and tools can change depending on the type of information page you're looking at. So as a gene protein page, you have the ability to, say, blast the sequence, or even look at the sequence. There's also tabs here to show different sub information about this particular gene information, protein information page. And I'm just going to show it all. We have lots of things that are provided, such as how the gene is transcribed and then translated, and whether or not it is a single protein or subunits within a complex. Reactions that it carries out are shown on schematics. We have Go terms that are available to help with classifying. So Go terms are gene ontology terms to help with classifying functions, molecular functions, the biological processes, or where in the organism the function is located. You can view things in genome browsers. There's a whole lot of genomic informatics that can be carried out, although we will be, I'm not going to focus on that today. Instead, what I want to show you is a couple other information pages. So here is a reaction page and showing the actual reaction that's carried out. We have atom numbering, which allows you to look at which atoms and parts of the molecules get moved around in the reaction. So that may be of use to some people. It shows what enzymes carry out the reaction, what pathways those reactions are in. Again, different operations available when you're on a gene page. For a pathway, let's look at the Calvin-Benson-Bassam cycle. And here, the pathways are shown and can be zoomed in and out, as well as changing the detail level. So zooming in and out, just making it larger or smaller. The detail level, you can look at different compounds in terms of their structures, or the enzymes that carry out each of those reactions. Again, you can zoom in and out. But the thing that all of these information pages have are interactive, clickable aspects. And so you could go back and forth between genes, reactions, even the compounds themselves, and get more information. Again, different operation panels that allow you to do things with, in this case, the pathway. One of them is to show on the cellular overview where that pathway is. And the cellular overview is essentially the metabolic pathways and reactions for the whole cell. We have, these also are very interactive and zoomable. The zoom takes a little bit of getting used to, so you don't go too much. Because we came from the pathway, it's highlighted, and we see it in two different places because, so the way we've structured, the way these cellular overviews or metabolic maps of the organism are presented is on the left are biosynthetic related pathways, in the middle are degradation related pathways, and towards the right are reactions that aren't associated with any specific pathway in BioCyc. Either they don't, it's not known what pathway they're involved in, or they're a single reaction. We also represent the membranes, and in this case, it's gram-negative, so it shows a cell membrane as well as the outer membrane with the periplasmic space. And you can see that there are transporters and some porins that are identified on the membranes. And reactions that might be carried out in the periplasm. You can do operations in here, such as highlighting things like compounds, clicking on by substring. You can start typing in, and it is a substring search. So as you start typing, things that start off with what you're typing show up. So if we want to look at, say, all the phosphate compounds or metabolites in the organism, we can do that and highlight it. You can also change the edge thickness so that you can see things better, interact by changing lights, color, list all the, say, compounds that are in the organism here, turn that into a smart table, lots of things that you can do to interact and explore. So that's a very quick look through the different kinds of information pages. Let me show you, and also how you could do a quick search, but let me show you there are also more object-specific searches that can be done that have a lot more options. So if we look at search genes, proteins, and RNAs, you could see there are a lot of different options by which one can search. You could type in a gene name like we had before, and choose that and look for it. But you can also look through by different properties, such as the molecular weight of protein, or its PI, or its gene ontology terms. You can even look to see what kind of evidence has been curated into the database. So whether or not there's information that's been inferred from direct assays. So which proteins might have that. So if we do that, click on that option and hit Submit Query. Oops, I think I still had... here we go. We need to click there. And no CBBL, submit query, and here we go. So here it just quickly put out the different genes that have evidence codes provided in them in the PGDB for Synechococcus elongatus PCC7942. So that's a quick way to look up more information. Okay, so that was a very quick pathway through navigation. And now let me get to the next part of the tutorial where I start showing you more of the tools. In particular, I'm going to start off with the SmartTables. And you can see here, turn into a smart table, this one result table that we came up with. So SmartTables are available in many places on the throughout BioCyc and within a PGDB. So if we go to the Tools menu, you could see SmartTables, and there are different kinds. So not only are there multiple ways of accessing them and making them, there are lots of different kinds. And I'm going to just show you the public SmartTables, which Peter already mentioned. This is a directory of SmartTables that many people have made. They're updated. The most recent one that's been put on is usually at the top. So with time, these cyanobacterial specific lists may not be at the top. And so it's good to be able to filter and look up what you're interested in looking at. So in this case, you type in the term that you want to keep, and filter out all the rest. And so not only are there those cyanobacterial lists, but also we have a list of all the cyanobacteria PGDBs within CyanoCyc and BioCyc. I see a couple things on chat just to, okay, so people saying goodbye, which is very nice. Okay. So those are the public SmartTables, but there are also, if you go back to the directory, there are my own SmartTables. So again, when you have an account, it's important that you go in through your account so that you could see whatever SmartTables you yourself have made and collected. And you have access to those. Special SmartTables are specific to the organism database that you're in. So if I click on that, we are in the Synechococcus elongatus 7942 database. So all these SmartTables, these special SmartTables are related to that database. And so things like all the genes within there are listed. These are provided automatically by BioCyc in order to make it clear as a convenience for people who are interested in that. Another way you can access some of this information is if you go to the information page for the organism. SmartTables for the genes, the polypeptides are also available here. Okay, so let me show you how to make a SmartTable. And so I usually start from my SmartTable directory. And over on the right, again, is that operations panel with things that you can do. And on here, you can actually make a new SmartTable. You could do it from an empty table, such as this. And you can add rows and type in information that you're interested in. For instance, if you're interested in ribulose, as you type, it's showing strings that are available within BioCyc. And so here, ribulose, we could instead take a protein, add a row, type in ribulose again if you want, and we could get to another protein. And what you see is it's converting it as you're entering it into an object that's within the within BioCyc. You could also just put in free text, but it's nice that it converts it automatically to the object, and you could just build a SmartTable that way. You can also upload a file to make a SmartTable. And as I was showing you previously, you can, I'll go back a couple pages. Oh, maybe not. Looks like it hung up there for a moment. You can turn some tables into SmartTables. And so there's always that little sign that you can do that. Okay, here we are. Yeah, I guess I was, oh, here we are. Turn into SmartTable. All right, so let's go up here and search and make a SmartTable and start working with the SmartTables. So let me see what I had originally decided I would work with. All right, so if we go and type in ribulose, you can, what happens when you type in a word like ribulose, it comes up with all of the search results within that database for that word, or that contains that word. And so you can see that there are 5 proteins listed, 9 compounds, scrolling down, the 3 reactions, all associated with the word ribulose. And each of these, you can actually turn them into a smart table. So I'm going to just turn the proteins into a smart table. So it's a nice small one that we could work with fairly easily, to show you some of the features of working with smart tables. So just to give you more orientation, we have up here the metadata with the smart table, where you can change the name to whatever you want to change it to. You could add a description. When you make a smart table, it automatically gets saved to your account. So you can find it again if you go to Smart Table directory. And the other thing that you can see, the operations panel, which changes slightly because we're now in a smart table. There are also different kinds of things that you could do to the columns, such as transform columns. And transforming, if I click this drop-down menu, are computational calculations or pulling out information computationally about the column that's highlighted. So in this case, it's just a single column of proteins. You can make a new column of transformed information, such as the pathways or the reactions catalyzed by the enzyme. So if I click on that, it will show those reactions that are catalyzed by the enzyme. We also have property columns that can, or properties that can be obtained and put into a new column. So a column of properties would be things that are, excuse me, which are within the object that we're looking at. So for instance, a protein has on its protein information page common names, session numbers, the location within the organism perhaps, or the gene sequence. Here, or in this case, it's a polypeptide. So this is information that could be pulled out and put into a new column. So here we have our proteins that we started out with, the reactions that they carry out, and the sequence of the proteins. Another thing you could do with the smart tables is make a new smart table if you want to have just the sequences. So you could hit this plus sign, and it creates this new smart table, starting with the first column being the sequences. It did keep the protein names, but now we've got a new smart table that's showing up with a new name, which you could change. You can also export this if you're interested, and since there's you could export it as a FASTA file or just to a spreadsheet text file that can be, opened in some other tool. Okay, so that gives you a little bit of information about using smart tables. You can also highlight other aspects, other columns, and carry out different transformations or properties for those columns. And so again, just like the operations on the right might change for each type of information page, some of the transform or property changes that could be made are going to vary depending on the type of object information is in the column. Okay. So let's use a smart table now to find out what genes are associated with pathways that have phosphate in Synechococcus. I'm not going to actually use WH8102, so I'm going to change the current database so you could see something we've already talked about. I've used this before, so it's a preferred database. I click on it, its name shows up there. I now hit OK, and it switches here, switches to the new database of Synechococcus WH8102. And it also switched to the summary statistics page for this curated database. I'm going to then search on phosphate. And the compounds, and turn those into a SmartTable because I'm interested in all those compounds. But I'm not interested in, one thing I actually didn't mention is you could see here there are, like in the pathways, there are different pathways, but there are also classes. And these classes of pathways is part of the underlying ontology, which it's a structured vocabulary on which the information in BioCyc can be organized, and it's a relational vocabulary. So classes is indicating the class of a type of, in this case, pathway. And if it's not a class, then it's a specific pathway that's within the database. So I had clicked on all the compounds that have phosphate, but they're also in this list of compounds classes that I don't really want. So I'm going to filter the SmartTable to get rid of those classes. And you can do that over here. There's the filter, and up when you left click on that, it comes up with a sub window for parameters to put in. And we're going to filter all rows that contain a substring. And I'm just going to say a case insensitive, so I don't have to worry about capitalizing. And I'm going to say class. And that should, and I'm going to put a colon behind that. So that's going to, we want to get rid of those. So delete any rows that contain the substring class in them. And when I hit go, it will do that. And I want you to note that there are 14 little pages of compounds, and a page has 25 of them on here. So if I hit go, it selects all rows. Well, that's kind of odd. Okay, let me try that again. Delete from this. Let me just do class. That should work. Oh, all right. When I try to do things on the fly, you see how you can get confused. So I think what I'm going to do is, I don't know why it's not, why it says it's going to delete all the ones with class in them. Maybe they're not in here. I thought they did come up. Okay, so let's not do that kind of filtering, but there is a way to filter out things that you don't want. You could also actually just click on different ones that you don't want and just delete those, select checked rows. Okay, when you do, so you could essentially, what I'm doing here by filtering is making new smart tables based on an existing smart table. And so this is things, we've already seen that by making it, clicking on a particular column, you could make a new smart table that way. You could filter out things. You can also, when you do transformations and adding property columns, you're making new smart tables. And like I said, all of these get saved automatically from the to your account so that you can use them and go to them later. Another thing that you can do is actually compare smart tables. Let me just check the, okay, more, thanks. Excellent. Is compare smart tables. So we have this smart table of phosphate compounds. Well, let's first transform it to look at the reactions that contain phosphate. That's going to take a little bit of time. Okay, there we go. And it's listing all the reactions with phosphate and then all the reactions associated with these other forms of phosphate. Well, I'm just going to click here to reactions of compounds and make a new table. And that now has switched even the reactions that were combined all together under one compound. It's now each row is its own reaction. I want to also get the gene from this. Oops, that's another transform. The genes of the reactions, and it's going to make a new column of genes that carry out those reactions. So now we have the genes that were that carry out phosphate, that carry out reactions that involve phosphate. But I'm not interested in all of those reactions. I'm interested in looking at smart tables of phosphate genes that are not involved in P stress. So what do I mean by that? Well, these are all the genes in the genome, but I have a data set of genes that were up and down regulated under conditions of phosphorus stress. And I can look at those genes in a smart table, and I've already made that smart table. So I'm going to show the directory, my directory, and here's Tetu et al. 2009, WH8102 P-stress transcriptomics data set. So I'm going to open that, and here are a list of genes that have changed their transcription in early and late phosphorus stress. So I know from the transcriptomics data set what genes have changed. But you might be interested in looking at, well, what genes didn't change? And it's kind of challenging to do that, except we have the ability with the smart tables to do set operations. And what I mean by that is taking this set of genes that we know have changed their transcription and doing a set operation. Give it a minute. Do a set operation with the table of all of the genes associated with reactions that are carrying, that have reactions that are associated with phosphate. And so what we're going to do is take the table of the stress genes, and let's make sure I get this set up properly. We're going to do a set operation, not a union or an intersection, but a difference. So it's going to be, actually, we're going to start with the other table. Let me go back to the directory. We have our reaction of compounds. So we're going to do the set operation from that. Okay, so the first smart table is this reaction of compounds that I made of genes associated with phosphate. And we're going to take the difference of this table using the table of the TET2 reactions. And yes, Amanda, I just saw your question. The transcriptomics data set is something I uploaded to my account. So it's not available on BioCyc public database. Here it is. So here's the, and I'll show you how to load that actually a little bit later, because I'm going to use this data set again. So here we've got now with this set operation, the first smart table, this one, with all the genes associated with reactions that contain phosphate. We're going to do a difference between this table and this set of P-stress transcriptomics data, making sure it indicates we're looking at the genes, we're doing the difference in the genes, and you hit go. All right, that happened. That was very fast. What we have now is a new smart table called the set difference of reactions of compounds from that first smart table and then the transcriptomics data set. Here are the merged genes from both of those, the reactions they're carrying out, and there's no data under early and late P-stress because these are the ones that did not have their transcriptome weren't up or down regulated. So we have now gotten information about what genes weren't affected under P-stress, if that's something you're interested in. So this is really just an illustration of how you can use these smart tables that are interactive, that can connect to the information, the structured information within BioCyc in the PGDBs, to then ask research-related questions. I've already shown you some of the other things you can do, and there's much more you can do with these smart tables, but I'll let you explore some of that. So, so the next thing I wanted to focus on, let me just make sure I'm doing time. Yeah. So the next thing I wanted to focus on is comparative analyses. And just like smart tables, there are lots of ways you could get at comparative analyses in BioCyc. And what I'm going to do is start off looking at this. So a comparative analysis allows you to compare either within the single PGDB, which we happen to still be in Synechococcus WH8102, or amongst multiple PGDBs, and you can choose those. So you can decide what kind of comparisons you want to look at and choose the different organisms. To choose organisms, you click on that button and up comes a page for choosing organisms similar to what we've changing the current database, but it allows you to select multiple ones. And you can do that individually by typing in names of things. So we could type in Synechococcus, and here is Synechococcus. Yeah. There's 6803. We could do Synechococcus elongatus. There's 7942. We could do Nostoc. So we can put in a variety of organisms. They get added automatically. And before hitting OK, you can actually save them to a new smart table if you think you want to use those again. So miscellaneous cyanos for comparative analysis. And save, and that then will show up in your smart tables as well. Another thing is you can actually go based on lists. So my lists, if you look at that, you can now choose different kinds, different smart tables that you might already have. And so I have miscellaneous cyanos for comp analysis. I've done that before. Here's one. I'm going to click on that and hit add. Oh, that's the one I just did. Let's do this. I have a lot longer list. So it's useful to be able to pull those in, and then you can hit OK. So we've chosen now the organisms that we want to do a comparative analysis with, and you can select all or just any of these, and then hit submit. And I'm not going to do it now just because it can actually take a long time. This was already done. I had that already finished. So it seems like it's needing to find it again. Well, I will hit submit here. And we'll see how long it takes. But it can take, for lots of organisms, it can take a while. So we'll let that run. And I'm going to open a new window of CyanoCyc. Change the database back to 8102. And show you some other ways, while it's running that comparative analysis, other ways to do different comparative analyses. So we looked at CBBL and the other strain. So let's look at it in this strain. Here's its protein, gene protein page. Here, under the operations panel, are all sorts of comparative tools and operations that can be done. So we can search for the gene in multiple databases. You could show orthologs in multiple databases. You could align them in the multi-genome browser or align them, their sequences. So I'll just show you the orthologs in multiple databases. Oh, how does it know which databases? Well, I had already chosen some databases, so it's assuming I want to look at it that way. And boy, we're having a little bit of lag here. Normally it's a bit faster. While we're waiting for that, I could see this page came up again. So this was that comparative analysis summary that I had made earlier. And is still taking a while on this page, but this showed up just so that overall comparative analysis results is really extensive because I had chosen all. So here's a table of contents of all the tables of information, such as database summary statistics, reaction statistics. We could just keep scrolling down, and here are the tables showing comparative summary statistics of genome size, the number of genes across all the organisms that I chose. And these are really super useful for just getting a sense of the information for different organisms. You can also so if we go into gene proteins, we could look at gene annotation. Yeah, why did that go away? Or let's do breakdown of pathways. And you could actually interact with these and click on them to look at more. So here we could show the comparison of each object within the amine and polyamine biosynthesis. for each of those organisms. So quite useful. All right, and here's that result again. So there was just a hang up in a little lag on the online. If we go back to that other comparative tool I just showed you before here, showing the orthologs and multiple databases, that came up. And we can see for the organisms that I chose on the list, the genes, the gene product, and then the operon for those. And here are some that don't have those genes annotated in the genome. Whether or not they don't have them, actually, we know E. coli here doesn't, because the gene I was looking at, which is CB, a Rubisco subunit, would not be in E. coli because it has to do with photosynthesis. Another comparative tool is Align in a multi-genome browser. And let's see if that's going to work or if it'll be too much lag. There we go. So this was shown by one of the speakers, and it's great to be able to see for all these organisms, the gene of interest is shown in the center, and then it shows the genome context surrounding that. And the colors are maintained for the orthologous genes. There's directionality shown. White are genes that are not found in the main organism that we started with, which was 8102. But it gives you a really nice snapshot of what's available and what's in that genome context for that gene. Another a way to do comparative analyses is if you go to these, well, there's cross-organism search. If you go to these search, object searches that I've mentioned before, at the top, you can search across multiple organisms. And when you click on that, you can select those organisms. It remembers what you've selected before, but you can also change those. So quite interactive. And sequence pattern search. Also, you can search for peptides, and you can search full peptides of other organisms. So it allows you to put in a particular sequence that you're interested in. Actually, that's a DNA sequence. I don't think I have a protein one, but you can put in a peptide or protein sequence. It needs to be within less than 20 characters, but then you can choose from SmartTables the organisms that you want to look at and compare across those organisms, looking for that particular sequence pattern. So what I've just shown you very briefly are multiple ways that you can do comparative analyses across multiple organisms within BioCyc. We allow those comparative analyses to go up to 70 PGDBs at a time, which is usually quite a bit and enough for doing, initial searches and maybe complete searches. Okay, so those are very brief, and I see it's already 12:30, but I did want to take, if you want to spend another 10 or 15 minutes to show you how to overlay omics data on the cellular overview. So I'm going to go back to cellular overview, which is that metabolic overview of the organism. And we're going to use the transcriptomics data that I mentioned before. And instead of just bringing it in from the SmartTable, I'm going to bring it in from a file. So if I click on Upload Data from File, another sub-window comes up, and you can choose a file. And I've already got it here. And it needs to be in a text format or CSV format. And all of these little question marks next to the different input fields provide you with information on how to input the information, how to input. So that's very useful because you're never going to remember everything I'm showing you. So you can look that up. We're going to bring in the data based on the gene names, and looking at the first column is the gene names, and that's column 0. And so we're going to bring in 1, 2 columns. And it's log twofold change data, so it's relative data, and we're not going to do any manipulations. And you can bring it in on this diagram. You could also bring it in as a pathway collage, as I've mentioned before, which we're not going to do that. But on the Omics dashboard, and that's just I'll show you what those two mean. So I'm going to hit submit, and if it doesn't take more than a few moments, there we go. Okay, that was fast. Excellent. So here we go. We've got our transcriptomics data that got overlaid onto the cellular overview. We can change the presentation a bit so it's easier to see. We can change the color scheme. I'm going to modify it to a 5-color scheme. And also, there are a lot of things that didn't change a lot. There's something funny with the font here. But so you can actually do proportional drags. I'm going to do a symmetrical drag. Oops. Okay, something's a little weird. Oh, here we go. Oh, no, there we go. There's some little glitch right now. But anyway, so you can modify how you want those colors to be split. You can make your edges thicker or lighter. What we could see, the upregulated ones, we have some red here. You can actually scroll in or zoom in and start figuring out, well, what's going on here? What genes are these? And so we could see the reactions that's involved in. You could scroll back out. Here's a transporter that is highly upregulated. So definitely interactive, and you could start examining what kinds of pathways and reactions the genes of your transcriptomics data set or other omics data sets are being affected in the overall cell of the organism of interest. Now, another tool that's related to omics analysis is what we call the Omics dashboard. And here, a new browser window opened up with the results. And the difference between the Omics dashboard and looking at the overlaying the omics on the cellular overview is Omics dashboard takes any of the genes that are up or down regulated and organizes them in a slightly different way. In this case, it's organizing them not just based on metabolic pathways of biosynthesis and degradation, but combining genes that are related to, say, energy or the central dogma, such as translation, transcription, or cell exterior. So it's organizing the data from a different perspective. And so it allows you to think and analyze your data in a different way. You can interact with these as well. You can move things around if you don't like the way it's set up. You can zoom in. In this case, I'm going to zoom in on cell exterior, what transporters are involved in this P stress, phosphorus stress. And here we see for transport proteins, there are amino acid transporters, ion transporters, what are other transporters? If you click around that data, it'll go into deeper into what makes up the other transporters. And I know it's hard to read here, but here are some urea genes that are involved. These are cyanate. So all of these, it gives you information about the specific genes, or you could, like I said, go back to the overall proteins, or even the, let's get this out of the way, or just overall categories. Okay, other things you can do, let's go back to the cell overview. You could actually invoke what's called the Omics data table for all the omics data, and it gives you all the information that we were just looking at, and you can actually download that. You can also print out things from BioCyc, and these prints are PDF formatted so that they are crisp and could be used for publications. Okay, let me see, what else was I going to show? Oh, the last thing is actually pathway collages. And if we go back to the tools under metabolism, is what's called pathway collage. And this was used a lot at the curation workshop. You can actually click on whatever kind of pathway you want to do from the organism that you're working with, or you can do it from the cellular overview. And so there are different ways to make it. So if we go to the cellular overview, actually, let me just make sure I'm doing this correctly. Yeah, no, let's not do it from the cellular overview. Let's do it just from here, invoking the, wait a second, am I in the right spot? No, okay. Maybe it was from the cellular overview. It's been a while. I've just learned how to use this. So, import data. Should have done this beforehand. I know what I'm going to do is I'm going to open up, since I'm right at the end, I'm going to actually switch over to my PowerPoint. You can hear my cat is meowing at me. There's the PowerPoint. Nope, wrong one. Oh, my apologies. Here is the tutorial. Down at the bottom here, pathway collage. Okay. And the reason I want to show this is because it's super cool. So it opens in a new browser, and you can rearrange the pathways and reactions and overlay omics data on it. And I forgot, you can actually add or replace omics data. So I opened up the pathway collage, and oh, I remember now how I did this. I took just 20 of the top upregulated genes and highlighted them on the pathway collage, and it opened up what pathways they were in. or what reactions they were in. So that's what these are here. And you can actually click and move on each of these. But what's even better is you can overlay the omics data by clicking on File and then Add Omics Data. And when you do that, a sub-window opens up that you can specify the parameters, and you click OK. And here, the omics data are overlaid, and you could see the genes and the amount of transcript log 2-fold change for each of those genes within the different pathways. So again, it's just another way to view and drill down and view and analyze your data on the reactions and pathways that are available in BioCyc. So, that was a very quick introduction to some of the tools that you could use in BioCyc, some of the bioinformatics tools. I don't know how it looks like several of you are still here, so that's really good. I'm totally happy to answer any questions if you have any at this point. I will be, like I said earlier, providing the recordings of this symposium, but I'll also provide my PowerPoint, which has, kind of guides you through going through some of these tools that I've talked about and that I've just scratched the surface of. As Osna mentioned in her talk, there are a lot of tools and it can seem a little overwhelming, but what you can do with them is so useful, and that it's worth exploring. So I hope all of you can check out BioCyc and CyanoCyc for your own research. And if you find it useful, I hope that you do, then you can interact with it more. And please fill out the survey that I'll send a link to after this. And I hope you enjoyed the symposium. So thank you very much. And again, if you want to write a question in the chat or even ask me right now, that's fine. I can wait around for a little bit longer. All right, well, I think I'm going to log off now. Again, thank you, everyone. Have a great rest of your day, wherever you are in the world.
Wait, Lisa, I think that there was a question in chat.
Oh, thank you. Is that Sam? I didn't see. Is it possible to upload metabolomics data? Yes. Yes, actually, you can. We support basically all omics analyses as long as it can connect to the data types that we have. So metabolomics data will connect to compounds. That said, there can be challenges with it, because if the output from your metabolomics data has names of compounds that aren't synonyms of names we have in the database, then you wouldn't see that. And so there can be, you may need to look into when things aren't matching, if it's because we don't have the same chemical name, but there are ways to do that. So yes, you can also combine, well, actually, I'm not sure if you can combine, I think, metabolomics data with transcriptomics and or proteomics, but maybe you could combine proteomics and transcriptomics as well. I can't remember now if you can do that. I actually am still learning about all the things that can be done with your data on BioCyc. But yes, you can do metabolomics, and I believe you can overlay it with transcriptomics data.
Awesome. Thank you so much.
Yeah. Let's see, were there other questions? I didn't see any other questions, I don't think. Okay. I guess I will sign off now. Thank you.